Yeah, it's a pleasure to be here to present to you, to introduce Eligochem, a company that was formed a couple of years ago, and to talk to you about polar drugs, which is the area of medicinal chemistry design and synthesis that this company is focused on. Got some quite interesting results that we've found. I'd like to take you through those, and very much looking for your input. And we are looking for collaboration around this, so any ideas you have, please approach me afterwards. Okay, just a quick bit of background around chemical space in drug discovery. There is a concept of drug space. There's a whole massive area of what's called chemical space, the possible compounds that can be synthesized. But you need polarity to enable a compound to be soluble, and also to get selectivity from the polar interactions tend to be more selective and tend to give selectivity to drugs. So that's a requirement in drugs. But you also need lipophilicity in order to get potency through lipophilic interactions and absorption. And there's a bit of a balancing act goes on between polarity and lipophilicity. And this is embodied in such things as the rule of 5, various rules about total polar surface area, rule of 3, etc. There's a range of kind of mantras that guide medicinal chemistry design, and there's this kind of region of drug space which balances polarity and lipophilicity. We've kind of felt this is a bit constraining, and actually medicinal chemists for quite a long time have been thinking, how do we get out of the box and actually expand the region of space that we can operate in? So there is an alternative concept that we've been looking at, which is what we call areas of kind of good polarity or good lipophilicity in this polar drug space. So there is some precedent, which I'm going to take you through, that some quite polar drugs, quite large drugs, are well absorbed, and there's a lot we can learn from that, and there's a lot we can use to apply that learning. So we're looking to expand drug space into this more polar region. So again, this is just a bit by way of introduction. This is a medicinal chemist trying to get across to a candidate from a lead, and they're in many cases in some projects just walking a tightrope between this aqueous and lipid balance, and we want to help them. Okay, why make polar drugs? There is another reason, which is to try and enhance drug safety. This is published information from Bioprint. Bioprint is where a compound is tested against hundreds of different targets, and you can look at polypharmacology. This graph shows the kind of proportions of polypharmacology. So only a small portion only hit one target. Many hit 2, and there is quite a significant number of drugs will hit quite a lot of targets in the Bioprint range. This is taken as a kind of surrogate for lack of selectivity, which is also, by extrapolation, a surrogate for potential safety problems or off-target safety problems. When you look at this Bioprint data and you look at this graph here, you've got lipophilicity increasing in this direction, and this is the polypharmacology. And you see a fairly marked effect. As you go to increased lipophilicity, you get increased polypharmacology. The same with molecular weight. When you increase molecular weight, you get decreased polypharmacology. Kind of makes sense. You've got a bigger molecule, more chance for specific interactions. But there is this downside to lipophilicity. And also, if you could make larger compounds, you'd be in a better place as well. So large polar compounds are perhaps the ideal place to be for safety from this type of data. And there's some other data of this type in the literature. Okay, so what about absorption? That's one of the main problems. That's what really holds people back when they start making large or polar compounds. And this is data from 480 compounds which have known human intestinal absorption data. And this is the number of hydrogen bond donors. This is NHs, OHs, which are the polar groups, the predominant polar groups in many compounds. And as you increase the hydrogen bond donor count, as you get up to 3, 4, as you get up to 4, you start to see a drop-off in absorption, and there's very little absorption when you get up to high hydrogen bond. This is into biological territory up here. But there is this kind of region of space. There are examples where there are compounds that are reasonably well absorbed with quite high hydrogen bond count. It's a minority of compounds, but they nonetheless exist. Similarly, this is just PAMPA data. This is passive absorption in a PAMPA assay. The same kind of picture. As you increase the hydrogen bond donor count, as you get up to 4, you get this plummeting of the absorption. But there are still compounds here. These are sort of the exceptions for passive absorption that are absorbed. So we tasked ourselves with investigating all these compounds, and in the literature there's about 50 or so compounds you can find that are anomalous. So this is a poster child. This is cyclosporin. And cyclosporin is quite a large molecule, over 1,000 molecular weight, quite polar, lots of hydrogen bond donors, but it is reasonably well absorbed. And when you look at this compound, and there's also other examples, this is atazanavir. It's again a reasonably large compound, quite polar, that's well absorbed as well. And you can rationalise these compounds through internal hydrogen bonding. So we postulate, and this is something which is fairly consistently observed for these anomalous compounds, that you can exist in 2 conformations. There's the open conformation in aqueous solution, where all the hydrogen bond donors are solvated. But some of the time, the compound can exist in a collapsed state, where it hydrogen bonds back on itself. It's a fairly unique property, and you see it's a more compact structure. It's on the surface, it's a more lipophilic structure. It's going to go across membranes more easily, and so you can rationalise quite a lot of compounds. There's other ones, Bazentan, aliskirin, and I say there's about 50 others in the literature that you can rationalise in a similar way. So again, just in summary, the postulate for this is you have a drug with hydrogen bond donors and acceptors. It can spend some of its time in an internally hydrogen bonded state, and that more lipophilic collapsed state is more favourable in the membrane. It can cross the membrane more easily. So essentially there's a reduction in the activation barrier to get across the membrane, and hence better absorption. Okay, that's the theory. We set about 2 years ago to investigate applying this in drug design. And what we did is we took just fairly standard parallel chemistry, privileged cores, privileged monomers, quite well known and established parallel chemistry, but we chose the cores and the monomers which had a high number of hydrogen bond donors. And we created a virtual library of about 100,000 compounds, and we then used a proprietary design technology which we derived from all the precedent in the literature to filter these compounds. And it's a 3-dimensional method for assessing the potential for internal hydrogen bonding. And we could then select out from this virtual library those which we would predict to have good absorption based on this propensity for hydrogen bond formation. And we synthesised about 500 compounds of this type. We've also looked at commercial compound space. There are about 6 million compounds out there that you can purchase, and we've done a filter of those using this methodology for predicting this internal hydrogen bonding. There's remarkably few compounds. Out of that 6 million, there's under 2,000 that have this property. So it's quite a rare property. But we've accumulated a portion of those now, and we have a library now of about 1,500 compounds, and we've been generating data on those designed and selected compounds. This just gives you a bit of an overview of that collection. This is a comparison with known screening libraries. This is a range of pharmaceutical libraries. This is from a publication in 2007, GSK, Merck, Pfizer. And this is molecular mass and C-log-P, and your typical pharma screening library, it's got a median C-log-P of about 3.5. It's around 420, 440 molecular weight on average. Our library that we've put together is relatively small. That was partly because commercial compounds tend to be small, and also we went for the easier chemistry, which tends to make smaller compounds. But the lipophilicity is about 2 orders of magnitude more polar than your typical library. that just gives you a bit of a distribution. Hydrogen bond donors, 4 and above. So between 4 and 6, the compounds we made, and quite a large number of hydrogen bond acceptors. Okay, now I've got some data. I can't show you the structures. These are proprietary compounds that were synthesized, but just trust me, they are drug-like and obey all the normal rules of avoiding all the unpleasant things that you can have in drug molecules. But we generally find that through doing, we've done pamper screenings. We looked at passive absorption, and we're generally getting good absorption. With hydrogen bond donors of 4, we're getting pamper fluxes of negative log of in the 5 to 6 region, which is quite a good flux. The most impressive data, though, we've got is with pairwise compounds. We've got a range of pairs of compounds where they're isomeric. They may have a hydrogen bond acceptor, it might be a pyridine, but the nitrogens move 1 around the ring. So, and that then means that it can't form one of the hydrogen bonds. And we've got now about a couple of dozen of these pairs, and we get a fairly consistent picture emerging that these are just 2 compounds. This one, they both have 4 hydrogen bond donors, but this one has the ability to form 4 internal hydrogen bonds. Its pamper flux is 6.8. The compound that can't form the hydrogen bond is 3 orders of magnitude lower flux. This is another example. It's a collection of 3 isomers, only one of which can form the 4 internal hydrogen bonds by prediction. And again, you get this 1,000-fold difference in absorption. So it's a fairly marked effect. Actually, this is bigger than we expected, but it's a fairly consistent observation. Now, the other thing we've done is we've looked at a typical screening library. I talked about commercial compounds, but this is a screening library you can purchase. It's ex-pharma. It's 100,000 compounds. We ran this through our 3-dimensional filter, and we found 78 compounds satisfy the criteria. And this is fairly typical. We've looked at several screening libraries now, and it's typically between 1 in 3,000 and 1 in 10,000 compounds have this property. An interesting observation is if you look at known drugs, about 1 in 10 known drugs have this property. That's just an observation, but there's an interesting, encouraging kind of observation there. So anyway, we were able to buy these compounds, so we were able to get our hands on 34, which we put through pamper. 23 of them had reasonable flux, which for selecting compounds of this level of polarity, where with this number of hydrogen bond donors, 4 to 5, we were quite pleased with. So I think we've generated at least an initial body of data suggesting that it's possible to apply this principle of internal hydrogen bonding to the improvement or the selection of compounds with good absorption in this polar drug space. And it potentially then allows us to step out from the rule of 5 and help this medicinal chemist. So the way we look at it, and very open for discussion on this, here's our medicinal chemist trying to get from lead to candidate, balancing these 2 properties, and we believe this approach widens the options, and we call it the amphiphilic superhighway. Amphiphilic is for dual property, that allows the medicinal chemist to progress much more rapidly through to the patient by giving this kind of wider window for managing the absorption. As I said, we're seeking collaboration. This is scientifically, it's been very interesting, but there's a lot more we can do in this area. We would like to go back and check that the polypharmacology really is better with these compounds now we've made them. We'd like to look at dermal and CNS absorption and actually look at more real life systems. We want to look at efflux and metabolism. Yeah, you might postulate that these more polar compounds would behave better there, but we'd like to generate some data. And particularly interesting, we'd like to look at PPI inhibition. The compounds I showed you were quite small we've made so far. We just recently synthesized a library of compounds up to 800 molecular weight, and we're seeing good pamper flux, actually very good pamper flux, quite striking. It does look like, as the literature would suggest, but we're able to proactively design compounds in that kind of 500 to 1,000 range, which is stepping us out further, which theoretically you might think would have more potential for PPI inhibition. Probably, because they're going to be more selective, the hit rate would be lower from any screening library. So we think that this approach probably has best potential for taking an existing lead and improving it, building on it, or taking very lipophilic compounds and actually making them more soluble by designing in an internal hydrogen bond. And because we've analysed all 6 million commercial compounds, there are several hundred scaffolds which lend themselves to this property. We're able to scaffold hop these into any lead now, so we can take any lead and very quickly come up with suggested analogues, which we would predict would either be more polar with absorption. So I'm very happy to talk to anybody about moving forward this science. It's been a really fun project over the last couple of years. I need to acknowledge Alex. Alex is a computational chemist, used to be head of computational chemistry at Pfizer in Sandwich. He is the brains behind this operation. He's defined and devised and refined the three-dimensional algorithm, and we keep refining that as we get more data. Graham Moore, medicinal chemist, he handles the operational side of Eligochem, getting the compounds made and the synthesis. Some of these more polar compounds do have their challenges. They tend to have more intermolecular participation. You tend to get lower success rates. So there is some chemistry tricks to this. But they're my other two partners in crime that have sort of made this happen over the last couple of years. And with that, I'd just like to thank you for your attention.
Thanks, Andrew. You're on time. Victor?
So, I don't have a microphone, but I have a loud voice. So if you look at the conformations of these molecules and compare them in aqueous solution versus in membrane environments, because you might have a change in conformation. We've seen this a lot with peptides. You have a different conformation in aqueous environments than you have in membrane environments. And if your hypothesis is right, then you should be able to also use NMR to detect that this is happening.
Yeah, I think that's a very good idea. I mean, that what you've just described is underpins the computational method. It's looking at a comparison between a solvated and a gas phase state. So that's. But yeah, no, I take your point actually.
It's a lipid state, not a gas phase.
Yeah,
state.
that's very good. So I think that would be a very useful experimental experiment to do. In fact, if you have any facilities to do that, we'd be very happy to collaborate with you.
Okay.
Any other questions?
You were saying you're looking basically a correlation between the internal hydrogen bonds and your C-log-P, or your actually absorption was quite favorable. But how does it correlate to the unsatisfied hydrogen bonds? Basically, when you eliminated the ability for one of them to form, and obviously you have some unsatisfied, is the correlation equivalent even if you just look at multiple unsatisfied hydrogen bonds? Does that increase the burden?
You get a fairly rapid drop-off. You know, with just one unsatisfied hydrogen bond gives quite a big drop in absorption. I don't think we've really analysed the data sufficiently to do the further extrapolation. It's a good question. Right. We probably do have data to support that, particularly with our compounds with 5 or 6 hydrogen bond donors. So I can take that away, but I don't have the analysis.
Obviously, this polarity or hydrophilicity is an important parameter, but is it in one way or the other also related inversely, for example, to metabolism, or directly related to urinary excretion? Because I'm wondering whether these properties you are studying in one way or the other can be extended to other ATME properties of the compound.
I mean, you might hypothesize that more polar compounds would be metabolized less. That's generally an observation. And you're right, more polar compounds tend to be more predominantly renally excreted. So you might hypothesize that would be the case. And yes, we would like to generate more data, particularly on the metabolism, because that's easier to do in vitro systems. So again, if anybody wanted to collaborate on that, we've got some great pairwise comparison compounds that can be used to do a fairly sensible experiment with a relatively small number of compounds to see whether you can see a trend in the properties. But that's definitely something we'd like to investigate.